So thank you for inviting me here. So I'm CELINE Molinaro. I'm CNRS researcher since only 2 years, let's say. And today I will talk about Plasmon induced light and heat for chemistry and biology. So first, so I come from Mulhouse, so it's IS2M, it's Institut de Sciences des Matériaux de Mulhouse. So the building looks like this. It's a pretty big institute. We are like 300 people working in there. I'm part of the smallest team of IS2M, so it's here. We are 4 permanent people working mainly in photochemistry. So part of the team are designing fluorophore for 2-photon absorption. We are designing many photopolymerization process at any wavelength, so from the UV to the infrared. Of course, we are doing plasmonics. And since I joined the team, so 4 years ago as a postdoc and now 2 years as a CNRS researcher, I introduced the thermoplasmonics thematic inside the team. And part of the team is also dedicated to green chemistry. So my talk will be divided in 4 parts. First, I will introduce what is plasmonics, which is the main idea of this talk. And then the talk is separated in 3 parts. So to see each time an effect of plasmon associated with an application. So plasmonic associated with chemistry to create hybrid nanoparticles for sensing. Thermoplasmonics associated with biology to grow a microorganism on a microscope. And finally, my actual research topic, so associating thermoplasmonics and polymer chemistry. So first, what is plasmonics? So what is a plasmon? So as soon as a light is shining on nanoparticles, so metallic nanoparticles, it will create a plasmon. So light is only an electromagnetic wave. So as soon as it enters in contact with the nanoparticles, the electrons of the nanoparticles will start to oscillate. And they will oscillate a lot. It's like an harmonic oscillator, and it will be a strong collective oscillation of the free electron of the metal here. These effects, so this interaction between light and nanoparticles and metallic nanoparticles can be quantified using cross-section. So basically, we have 3 cross-sections. So the absorption cross-section, so it's the particles that is absorbing light. It can scatter light, so re-emitting by scattering. And we can add these 2 to have the extinction cross-section. And these 2 terms are dealing with alpha, which is the polarizability, and which is interesting here, because as soon as we want to maximize the collective oscillation, we need to maximize absorption cross-section and scattering cross-section. So we need to maximize this formula. And to maximize this formula, we have to set this to 0. So we need to have the permittivity of the nanoparticle equal to -2 the permittivity of the surrounding media. And this occurs at a precise wavelength, which is the Plasmon resonance wavelength. And basically, if we look at the absorption cross-section of a gold nanoparticle, so here it's a sphere inside here, so we have a resonance here, which is this peak at around 532 nanometers. So this is the Plasmon resonance. At this wavelength, the conduction electron of the metal are oscillated at their maximum. So what does it mean? So if we have a nanoparticle here, which is interacting with field lines. So if we are outside of the plasmonic resonance, nothing happens. I mean, the lines are just here around. But when we are at the Plasmon resonance, the interaction of these nanoparticles with the incoming light is way wider than the nanoparticles itself. So this is the plasmonic resonance. So plasmonics, so in science, plasmonics exists really since 70 years. But it exists, it has existed for over more than 1,000 years through this object. Maybe you know this object. It's a Lycurgus cup, and it can be red or green. And this is due to plasmon. in this case, the cup is illuminated from inside. So we observe the light which is transmitted. So this cup absorbs green. So if we shine the light in reflection, it will appear green. And this is the first plasmonic object that was ever created. Of course, at this time, they were not doing any chemistry to synthesize the particles. They were just reducing metallic powders such as gold or copper, and they mixed them up, and they efficiently create this. So now I will focus on the plasmonic resonance. So of course, the plasmonic resonance will vary with the metal that is used in the nanoparticle composition. Here we have an example of two colloidal suspension of particles. It's like the same particle. It's a sphere of 30 nanometers, but here it's gold sphere, and here it's a sphere made from silver. So their color are different. Here it's red, here it's orange, and meaning that their plasmonic resonance is different as well. So we can tune the plasmonic resonance with the metal inside the nanoparticles. Another way to tune the plasmonic resonance is to play on the size of the nanoparticles. Here it's gold, but with particles of different size, so with increasing size. And we observe that this solution is more reddish than this one. If we observe their plasmonic resonance, we observe a slight shift of only 8 nanometers. If we perform some simulation of a cross-section extension of these different particles, we will observe the resonance redshift, which is really small, let's say, but we observe a strong increase in scattering and absorption due to the size of the particle. Of course, today we can do chemistry to synthesize all of these particles. So we can create spheres with resonance in the visible, so at 532 nanometers. So in solutions, they appear red. We can synthesize rods. Rods are particularly interesting because they display two resonances. One here, around 532, which is due to their transverse plasmonic modes, and one here, which is also tunable, and I will speak about this later, which is due to the longitudinal resonance. And if we synthesize triangles, it will present another resonance as well. So let's get back to the rods. So if we increase the aspect ratio, so the length of the rods, we will observe a different behavior in terms of plasmonic resonance. For small rods, the suspension here appears blue, so it absorbs here in the red. And then for longer rods, it will the plasmonic resonance will shift to the infrared, and the solution starts to be red. So with this, we can address all the spectra of plasmonic resonance. And the last one, which is interesting, is the fact that the plasmonic resonance is also dependent on the surrounding environment. So here, I plot the extension cross-section of a sphere in air, so with a surrounding environment with a refractive index of 1. If I plot the same, but for particles inside water, so with this refractive index, it will shift the plasmonic wavelength. And it shifts all with, it shifts to the red when we increase the refractive index. And this is interesting for sensing. It's able to detect any refractive index change at the nanometer scale. And we have also an increase of the extinction here. So just to summarize this section, so I'm sorry, the figure is in French, but it was a really good figure, so I put it in French. So we can tune the plasmonic resonance with the metal nature of the nanoparticles, with the shape, with the size. I didn't talk about this, but if we put many nanoparticles inside and together, and if they are close enough to interact between them, we can also tune the plasmonic resonance. So this is interesting, but what happened after the plasmonic generation? So for now, I just talk about what happened here. So the plasmon is generated. We shine light on the nanoparticles. We have a strong oscillation of electrons. But what happened after this? So in a really short time scale, we will have many effects that are interesting. First, we will have hot carrier generation. So it's energetic electrons which will come across the nanoparticles. And then they will rapidly thermalize in the time scale of picoseconds. So it's really, really short. They will thermalize through Joule effect. You will have a thermal dissipation, so heat which is created. So from these particles, we can expect a strong field enhancement. So it's what I described before. We expect electron hole pairs, which can be used, and heat. And all this effect find many applications in plasmonics. If we look at what we can do with the field enhancements, so we can do luminescence microscopy based on gold nanoparticles or other nanoparticles. We can create nanolasers, nanoemitters. If we put fluorophore at the right place, close to a nanoparticles, and we can, due to the strong field enhancements, we can create optical tweezers to trap any objects here, so nano objects around the nanorods, for example. We can use the electron hole pair in photovoltaics to enhance the yield of solar panel. Plasmonics has many applications in photocatalysis. And heat is the most famous application of heat, so it's phototherapy. We can, based on chemistry, we can put nanoparticles inside cancer cells and shine a laser on that. It will create hyperthermia locally, and the cancer cell can be dead then. Of course, it has other applications, for example, in photovoltaic. So just before going to the next part, so I don't know if you know Mentimeter, so it's not maybe as fun as the one we have this morning, but if you can join here with the QR code, we will have like a two question test on plasmonics. So let's start it. So the first question is this one. So why are gold nanoparticles preferentially employed in plasmonics application compared to other metals? So the results will be displayed instantly. I can see that you really listen to my talk, because a huge answer about plasmonic resonances visible is one of the good ones. So basically, we can use a lot of metallic nanoparticles in plasmonics, so silver one, copper one, and gold, of course. So gold is preferentially employed due to its chemical stability against, for example, silver. Silver will oxide rapidly, so it can be used, but it's different. So it has a resonances visible, which is really interesting. It's biocompatible, so for biological applications, that's great. So there is a control of the shape for gold nanoparticles, which tunes the resonance wavelength, but it's not exclusive. If you use silver particles with different shape, you will tune the resonance wave as well. So it's not really expensive due to the fact that it's gold. I mean, it's expensive as silver particles or others. The next question is this one. So among the following images, which one depicts the plasmonic phenomenon? And there is only one correct answer. So let's check the correct answer. So the correct answer is this one. It's here. It's stained glasses. So it's made of plasmonic nanoparticles. So it's really old technique. So it's like the Lycurgus cup. The butterfly wings here, it's due to photonic crystals. So it's not plasmonic. There is no metal inside. This is molten lava, so it's only the thermal radiation due to the temperature, so it's not plasmonic as well. And here it's a rainbow, so it's a refraction of the light in water droplets. So this is not plasmonic. So thank you for this participation. So let's go back to the presentation. So now you know all about plasmonics. Let's try to observe what we can do when we made chemistry interacting with light induced by Plasmon. For the light, when we shine light on a nanoparticle, it will induce a strong field enhancement. So the nanoparticles will act as a nano antenna. So it's like a classical antenna, but at the nanometric scale. For example, here we have the field enhancement around the triangles. So the field enhancement is located at the tip of the triangle here for a given polarization. So it gives like a directivity of antenna. If we change the incident polarization, this plasmonic hotspot doesn't exist anymore, and we have two others plasmonic hotspots here at the two other tips of the triangles. Of course, this field enhancement is different between nanoparticle shape. It will be different when we use different wavelength. And the intensity is a way difference between two nanoparticles. When we can put chemistry inside this to map the intensity field area by polymer chemistry mainly to create hybrid nanoparticle. Here the goal is to position at the nanometric scale a molecule where the field is the highest. So why we want to do that? We want to do that for a sensing application. Because if we put the molecule here where we can detect it the most efficiently, we will win a lot of detection sensitivity. So this has clearly good application in detection and sensing. So how to create this hybrid nanoparticles? So to do that, we take a lot of different nanoparticles, so with different shapes, which were synthesized by our collaborators, so Sylvie Marguet in CEA and Guillaume Laurent in PPSM. So this, I didn't mention it. This is a work that I carried out with Amin Kitous, which was a PhD at IS2M. So we took nanoparticles, we put them on a transmission electron microscope grid, so it's the blue items here, in order to have the nanometric resolution when we image them. Because here you have transmission electron microscope images of the nanoparticles. So with this, we can observe if we have or not polymers around our nanoparticles. So we put the nanoparticles on the same grids, we put formulation on top, we shine light through laser illumination, so we can control the power, the time, and the polarization of the laser. So it was a laser in the, at 800 nanometers. So we shine light on that, and then we develop the structure. So if polymers are if the formulation polymerizes around the nanoparticle, we will see it on TEM imaging. And to do that, we use photopolymerization, because inside the chemistry, we can address different phenomena. And here we want to map the field enhancements, so we used a photopolymerization formulation. So basically, we just mixed a monomer here with a photoinitiator. So the photoinitiator absorbs at 400 nanometers. So with this, we can perform 2-photon polymerization. Before doing any experiment, we have to know the threshold dose of the system. So it's the dose at which the system will polymerize in the far field. What we want is to observe photopolymerization only in the near field, so what is enhanced by the nanoparticles. So we need to be under this threshold dose, just to have the polymer starting here and not on the outside. To do that, the reaction is based on the photobleaching of oxygen. So you have oxygen everywhere. The oxygen will not allow the polymerization to progress far away from the particles. So what do we observe on gold spheres? So it's disk, it's not sphere. So here we observe that for a small threshold dose, there is nothing around the nanoparticles. So this is an important image. It means that when the polymerization did not occur, no polymer at all was on our nanoparticles. Here we observe polymer, here, here, and here. So we observe a ring of polymer from 2 to 5 nanometers around the nanoparticles. And we have an homogeneous polymerization, which was not expected due to the spatial distribution of the field here. So we expect for a given polarization to have a polymer lobe here and here, and not an homogeneous polymerization. If we took these nanohexagons, so again, for the smallest power sense, we observed nothing. And for increasing power, we observed a ring of polymer around. So a polymer shell, which is around 6 nanometers in thickness. And it's, again, homogeneous. It's, let's say, slightly anisotropic, but not that much, compared to the spatial distribution of the field here. So here, for these 2 nanoparticles, we were not able to map efficiently the field distribution of our system. So let's take the 2 other nanoparticles, which are more, let's say, anisotropic. So here for the rod, we observed, so nothing for this dose. And then we observed 2 polymer lobes here located at the tip of the rod, when the polarization is along the long axis. When the polarization is along the transverse axis, we observe nothing, which was expected this time. Because here when the polarization is along the long axis of the rods, we are at the resonance of the nanoparticles. So we have strong field enhancement at the tip. Here, when we are orthogonal to the longitudinal axis, there is no resonance. The resonance here is located at in the visible in the green. So here we have a wider polymer thickness, so it's 8 to 12 nanometers. And we have a change with the incident polarization, and we have a strong correlation between this field map and these experiments. When we go back to our last object, so the triangles, here so we still observe nothing, and then we observed polymer lobes that are located in the field direction. So here the field is along this, so we have two polymer lobes. When the triangle is oriented slightly different, here the field is along only this tip, and we observe polymer only here, which is again clearly expected. So in the field map, we observed here a strong field enhancement at the tip of the triangle here for this polarization direction, which correspond to these experiments. Here the field is along here, so we have the two lobes of a field, and we observe them here. So here again, you have a correlation between the simulation and the experiments. So why? It was not working here, but it's working here. So when we plot the field contrast of this, we observed that the intensity, we first observed that the intensity for the gold nanorods and the gold nanotriangles are much higher than the one from the nanodisk and the nanohexagon. And we observed that the contrast is not really good for these two objects. So here the field contrast induces more or less a chemical contrast, which can be explained by a diffusion of radicals from the area with high fields, or from a diffusion of oxygen, which is lower, which will quench the reaction. So it can be explained like this. But if we look globally to these results, we are able to put polymer where the field is the highest for some particles. And thanks to that, we apply this to concrete experiments with detection. In our lab, we are able to create molecularly imprinted polymers and to put them in formulations that photopolymerize. So molecular imprinting is a technique that creates specific reconnection sites for a target molecule. So the target molecule is in yellow here, which is also called a template molecule. And these functional here monomers and the cross-linking monomers are copolymerized in the presence of a template in a solvent. So this template can be the target molecule or a derivative. So the functional monomers here, they are initially form a complex with the target molecules. Then it will all be polymerized here. And the cross-linking is rigid enough to really stuck the target here. So then when we remove the imprint here, we will have a hole here, which will be able to recognize the good molecules due to the fact that the cavity, which is now empty, has the size, the shape, and the chemical functionality complementary to those of the target molecule here. So we have a specific reconnection site for a target molecule, and it's as selective as antibodies. So sometimes molecularly imprinted polymers is referred as plastic antibodies because it's the same behavior. So to do the experiments, here we forget totally about the individual nanoparticles synthesis. We need to have a lot of nanoparticles to do these experiments. So basically, we evaporate gold on glass layers. We heat them at 400 degrees, which will dilute the gold. So we will have a sample here with a lot of nano islands. We perform photopolymerization with a formulation with molecularly imprinted polymers. So we have this. So we have the nanoparticles, the film around here, and the target here, which was methylene blue. So when we rinse these samples, we remove the target, so the methylene blue, and we can incubate it to check if we can detect methylene blue. So to detect the chemical signature of the molecule, we use the Raman spectroscopy. So basically, a Raman spectroscopy provides the access to the vibrational level of molecules, so it's like their signature. And here, for example, this here is the signature of the methylene blue. And here we can see that for increasing concentration of methylene blue, we observed an increase of the signal. But what is interesting here is to see that we can detect with this sample 10 nanomolars for methylene blue here. If we want to check that this interaction is specific, we can do the same experiment, but instead of putting molecularly imprinted polymer, we use non-imprinted polymer. So it's the same thing, so the same functional monomer, the same cross-linking monomer, but without the target. So the specific recognition sites were not created. What we observe is we can detect here, so with the non-specific interaction, the methylene blue, but at higher dose. So clearly here, it's working as a molecularly imprinted polymers. But to create a good sensor, we need to have a short analysis time. So it's the case here. In less than 5 minutes, we can observe the signature of the methylene blue. So it's quite a short analysis time. And in terms of robustness of the sensor, if we do 20 cycles of incubation, rinsing, we can still observe the same plasmonic resonance, so here. So it means that in the vicinity of the nanoparticles, the polymers had not changed at all. And if we observe the Raman signal, it's the same after 20 cycles of incubation, and it's the same here after 20 cycles and after only one cycle. So as a conclusion here for photopolymerization and plasmonics, we were able to graft polymers where the field is the highest, so here at the vicinity of the nanoparticles, here, where the plasmonic hotspots are. It's effectively mapped by the polymer for these two, so for the rods and for the triangles. And when we combine these two molecularly imprinted polymers, we can create sensing platforms that are fast, robust, and selective. Of course, here I present only results about methylene blue, but we tried other molecules such as biological molecules, or it can also work with PFAS. And we have a really good detection limit so for the methylene blue. So let's now go on the next part. So it's about thermoplasmonics. So it's not the same effects, but I will describe it and how thermoplasmonics can interact with biology. So the thermoplasmonics is the last effect that is induced by the plasmon. And it will just create heat. So it's like a toaster, it creates heat. So the famous application is phototherapy, where it can basically kill the cancer cell. But here I will present another application. So I worked on that project during my second postdoc when I was at Institut in Marseille. And the goal of this project was to observe thermophiles in vivo on a microscope. So I carried out this research for more than 2 years in Institut Fresnel with Guillaume Bafou on his ERC project I4. And then I will talk about my current topic of research, which is dealing with thermoplasmonics, but chemistry with the polymer. So we will now talk about biology, and you might have known that life exists in a broad range of habitats. meaning that we have microorganisms that are called extremophiles that are able to live in lake like this, where it's extremely salty. And this species, the microorganism that is able to live here, give the pink color of this lake. In acid lake, it's possible to find microorganisms that is called Picrophilus torridus that live at pH close to 0. If we look at where on Earth the temperature is high, so in black smoker at the ocean floor or in geyser in islands, we can find microorganisms that are called thermophiles that are able to live at temperature as high as 120 degrees. Of course, you can find microorganisms that mix many extreme environments. So here you have an archaea. So I will explain this later, what is an archaea. So this polyextremophilic microorganism, so Sulfolobus acidocaldarius, needs an acidic pH and a high temperature to live. We even have a bacteria, so Deinococcus radiodurans, which is able to resist to the cold, to the dehydration, to the vacuum, and to radiation. So here I will focus on thermophiles, because the goal here is to do thermoplasmonics. And there are two different microorganisms which are thermophile, which are the archaea and the bacteria. So what is an archaea? Maybe you have learned in biological courses that you have two different kinds of cells. You have the prokaryotes and the eukaryotes. So the prokaryotes, inside, you have the bacteria and the archaea. It's single cells. And here you have the eukaryotes, like plants and animals. And if we look at this, so it's not a two-domain system, the biological classification. It's a three-domain system. So we have eukaryota, bacteria, and archaea. And archaea have characteristics which come from the eukaryota, like the genetic and metabolic pathway, like translation and transcription of the DNA. But archaea has present also share also behavior with bacteria, like circular chromosomes and the morphological similarity. It's smaller than eukaryota. So let's go back on thermophiles. So thermophiles need at least 50 degrees to live, so we can find them in the geyser, in a hot spring. Sorry, it was not translated. And we can also find hyperthermophiles that need 80 degrees to live. So biologists are already studying them for like many years, but they need autoclaves to replicate their living condition. And an autoclave is nothing more than a pressure pot. So you cannot see what is inside. So you cannot observe them living. So that's really bad for a biologist to observe a microorganism only when it's dead. So they were able to do only post-mortem observation. If we look at the state of the art of how to observe thermophiles in vivo, we can find only 10 papers dealing with this subject. But it's like pretty old, let's say, paper. It was like 30 years ago. They built the first high temperature control observation chamber, which was looking like this. So basically, they eat everything. They eat the microscope and the detector to see it. So it's really huge. It's really, let's say, dangerous because everything is hot and it's not really stable. More recently, they designed smaller observation cell, such like this, which combine with an objective which is not heated this time. They observed the DNA here divided in real time for this archaea, so Sulfolobus acidocaldarius. Another group, so here with a system which is not the same but comparable here, they eat the whole culture here of microorganism, and they were able also to observe the division of the cell here. But these two studies were focused only on Sulfolobus acidocaldarius live imaging. And they were constrained by many things, so they can only have at their maximum 80 to 90 degrees on a microscope stage. We can ask question of safety because everything like is really hot. It's time consuming to achieve the target temperature. So it took like hours to have their temperature here in order to perform this resolution. So the goal here was to design, based on thermoplasmonic, a system that allow the in vivo imaging and observation of thermophiles. So the system was this one. We have glass samples on which we have nanoparticles. We shine a laser on this. So based on thermoplasmonics, it will hit the system. And we combine this with a light probe here to know the temperature. So the gold sample was looking like this. And the system is working as following. So we have the laser rating. It will hit the nanoparticles. The nanoparticle will hit the environment, which is water. Upon this, the water, the refractive index of water is dependent on the temperature. So this can be detected by the light probe through a thermal lens effect, which will induce a wavefront distortion, which can be imaged with a phase camera, which is also called quasi-wave lateral shearing interferometry. And we observe this. And thanks to post-processing and a well-developed algorithm, we can go back to the temperature on the sample here. So with this technique, we can have the temperature of the sample. We can reach up to 200 degrees. We have a high resolution due to the objective we can use. It's fully customizable for fluorescence. The temperature is reached instantly. But of course, it has also drawbacks. In order to avoid any convection in the media, we need to have lamella, here a glass covered slip on top, and a layer thickness which is less than 20 microns in order to avoid any convection. And we can ask the question of phototoxicity due to the laser hitting here at 800 nanometers and induced also by the light probe here, which was set at 600 nanometers. Of course, all of this can be tunable. So the experimental setup was this one. So we have the sample here, which is hitting by the laser. The laser was, we can change the shape of the laser beam due to a spatial light modulator. It was sent here on the sample. Then the LED here was used for the wavefront distortion and sent on the camera. So at the scale of the bacteria, let's say we are like this. We have the laser here, which is shining light on only some bacteria, and we want to observe them alive. So the first experiment that I carried out was on Escherichia coli, so a well-known bacteria. I sent the laser beam on the sample, so I imaged this with the camera. So I know that at the center of the beam, I have 37 degrees. And here I observed the growth of some bacteria here. This bacteria is growing. The others are also growing, but sometimes when they are dividing, they are like ejecting the divided cells, so we cannot see it. Here I observed with the camera a doubling time of around 25 minutes, so it confirmed a temperature of 37 degrees, which was already acknowledged by the camera. So clearly here it validates the technique, but not for thermophiles, for Escherichia coli, which is like a mesophile. When I tried to do the first experiments with thermophiles, it was, let's say, really hard. Sometimes in research, even if you validate the technique, it doesn't work. And in biology, it can come from lots of parameters. Here, it was due to the fact that I was trying here to observe the growth of Geobacillus stearothermophilus, which is a bacteria, thermophilic, which is strictly aerobe, so it needs oxygen to survive. And I had to deal between the need of oxygen and the need of the spatial confinement in order to avoid convection. If I remove here the glass, of course the oxygen can diffuse inside, but I will have convection and I cannot see anything. So I carried out some experiments to check that if the problem comes from the oxygen. So here, I hope that we can see something. Here, it's two bacteria that can be facultative anaerobe. So if they have oxygen, they are okay with that. If there is no oxygen, they are also okay with that. They can grow. So here on black, I have the cover slip and the time here. So for Escherichia coli, it can grow under the cover slip and outside. For this bacteria, so it's a lactobacillus like you have in the yogurt, it can grow under the cover slip as well. But if we look at two thermophiles which are strictly aerobe, so Thermus thermophilus and Geobacillus stearothermophilus, if we look at what happens under the cover slip, there is nothing. It cannot grow in a small environment. Not because they are claustrophobic, but because they need oxygen to survive. So we had to change the setup here and to design another observation cells with a top glass here with a hole. And all the experiments that I will show after will be performed close to the hole in order to have oxygen to diffuse inside the bacterial culture. And when I did that, here is what we can observe in the video. So at the center here, I have more than 60 degrees, so a temperature that allows the growth of Geobacillus stearothermophilus, and we can clearly observe in vivo the growth in real time. Of course, and it is directly linked to the laser on. I don't know, I will put the video up from the beginning. So here the laser is off. When I turn the laser on, the bacteria starts growing in the 10 minutes after the laser is turned on. And when the laser is off, the temperature goes back to the ambient temperature, which is like 20 degrees, and the growth of the bacteria stops immediately. And when I return the laser on, it will go on. It's another example here with a different, it's the same bacteria, but with a different lasers. And here again, we observe the growth inside the laser beam, where the temperature is high enough to allow the growth of this bacteria. What is interesting with this system is that we have the phase camera that allow us to know the temperature, but we can measure the increase of the biomass here, so it's the cells, of the different colonies from initially isolated bacteria, like we have here, within the field of view of the movie that I presented before. And the dry mass measurements is performed with the same camera as the one that gives me the temperature. So if we plot the dry mass with the time for different temperature, we observe that for higher temperature, we have a strong growth, which is totally expected. And as expected, we have a temperature here. Oh, sorry. We have an exponential increase of growth here, which is depicted by the semi-log here. And we have and at the end of the experiments, we reach a plateau here, which is due to the fact that we have many, many cells, and it happens also for biologists where they are doing culture. It's called it's called stationary phase. If we plot the growth rate at a function of temperature, we find out that this kind of curve, which is a curve that was already found by biologists when they use autoclaves. So here I have another movie of the cells, because Geobacillus stearothermophilus is also able to live, to swim. Sorry. So here the laser is turned on, so nothing is moving on the images. It's turned off, sorry. It's a real-time video, so that's why it's a bit long. At some point, the laser is turned on, and we can observe a bacteria here, which is swimming. And we will observe it here again. So it's real-time activation of thermophiles. So this bacteria, so here it's still a Geobacillus stearothermophilus. It has ability to form resistant spores when its environment is unfavorable. So in extreme condition, in cold condition for this. And it's like a mean of protection. So the spore can germinate and produce again cells when good conditions are back again. It's a well-known process by biologists, but it was never observed in real time. So here we have the spore of the Geobacillus stearothermophilus. So it's a zoom on them. Here they are in between the two temperature gradients. So they are at around 55 degrees, and we will observe them growing again. So the spore, it's like small balls. So it was the first observation of the germination process in real time. So it's a great tool for biologists if they want to study this process. And of course, here I show you only one movie at a time, but it's like it's many experiments that were working. So then here we validate the setup for thermophiles, so for Geobacillus stearothermophilus. Then we tried to observe Sulfolobus shibatae, which is an acidophilic thermophilic archaeon. So it needs 80 degrees to live, and it needs an acidic environment to live. So here you have one. Here it's not anymore a long bacteria, it's a cocci. So that's why it's more small, and it's spherical bacteria that are dividing here. And of course, here we also extract the dry mass measurements, and we plot this as a function in time. And we observed again an exponential growth, which is expected, and a stationary phase when the cells are too many at the same place. So clearly, we can use thermoplasmonics to observe thermophiles growing in vivo. So we validate the setup with the bacteria, which needs 600 degrees to live. So we observe the growth, we observe the swim, and we observe the germination of the same bacteria. It was also validated for hyperthermophiles, which need 80 degrees to live. So clearly, it's a great application of thermoplasmonics that maybe will help biologists to study this organism in the future. Let's go back on the quiz. I have just one question. Yeah. So I think you are connected, you are still connected on that. I can go on. Or maybe I will let you 2 minutes. So there is only one question here, and it's this one. So in thermoplasmonics, so using the setup I previously described, what is the maximum of temperature that can be reached? So here we have like many different response. So the good answer is 200 degrees, which is surprising, because we all know that when we want to cook pasta, for example, we need 100 degrees to have the water boiling, but not on this system. It is due to the fact that the sample is really flat and water needs nucleation sites to start boiling. That's why when chemists want to have their experiments boiling in water, for example, at 100 degrees in their flask, they use a stone, which is, I'm sorry, I just have it in French, which is called pierre ponce. So it's a specific stone that will create nucleation sites to have the water boiling in the flask. But here, the sample is flat. It's like nanometric gold nanoparticles on a really flat surface. So we can achieve 200 degrees on the substrate, which is really interesting to address the question of temperature maximum of life or things like this. So now I will talk about the last part of my talk, which is dealing with chemistry and thermoplasmonics and to create thermal probes based on polymer. So here I still have 3 effects of plasmonics. So the field enhancement. In the second section of my talk, I showed you that we can put a polymer here where the field is the highest using field enhancement. We can graft polymers using electron hole pair through another chemistry, which is called diazonium salt. So I will not talk about that, but I present you the fact that it exists. And it is located where the field is the highest as well, but it's where the electron hole pair are generated. And the heat can be also used to graft polymers on nanoparticles, and that is what I will explain here. So I started this field of research trying to create hybrid nanoparticles based on the grafting of polymers with heat on a substrate. It has the advantage of using other chemistries and photochemistry. It can be used to create temperature probe at the nanoscale. So here we are going back to chemistry, but let's say it's simple chemistry. It's still polymerization. And I mixed only 2 components. So I have my monomer, which was the same as the one used in photopolymerization. But here, I replaced the photoinitiator by a thermal amorcer, so which is called diams, and which is synthesized by a team at ICR Marseille. So it's not photopolymerization, it's really thermopolymerization. So if we observe the spectra, so the absorbance spectra of this formulation, there is nothing, there is no absorption. We cannot do photopolymerization with this formulation. And if we observed the heat flow, we observe that this polymerization, this formulation, will start to polymerize at 130 degrees. The mechanism is pretty simple. We have the thermal initiator here, which, and when it will reach 130 degrees, it will break and create radicals. These radicals will interact with the monomer, PETIA, and then the polymerization starts. So I validate this tool through this experimental setup. So I have a sample here made of nano-islands, which were created by thermal dewetting. I put the droplet of polymer here, and I close the sample. Then I send a laser illumination on these samples at the plasmon ring resonance. It will hit the substrate. Then I will open my samples, rinse with ethanol, so it will remove the unreacted formulation. And if we observe polymer, it means that the temperature was exceeded on the sample. So my samples is this one. So it's a random network of nanoparticles. It has a resonance wavelength at 570 nanometers when it's covered by formulation. So here it's just a shift of plasmonic resonance due to the change of refractive index between air and formulation. So to address it to address it efficiently, I use the laser illumination at 532 nanometers. And I obtain that. So here is my sample before experiments. Here it's for power that did not induce any polymers. And when I increase the power, I observe polymer dots here, which are increasing in size. Of course, if I illuminate the same sample but without gold nanoparticles, there is nothing. So there is no photopolymerization. It's only thermopolymerization. And we observed like really huge, let's say, polymer dots, because there are like 70 microns in thickness and several millimeters in diameters. This can be surprising due to the fact that we have nanoparticles that are generating the heat. But when we look at the theory, it's normal. So to create temperature on a sample through thermoplasmonics, we have two behaviors, which are adding, it's a sum of two behaviors, a confinement regime and a collective regime. In this regime, so the confinement regime, it happens, So in the confinement regime, the temperature increase remains confined at the vicinity of each nano source of heat. So here we have heat only in each nanoparticles. It happens for nanoparticles with low density, so in substrate with low density of nanoparticles, when the illumination area is small and with continuous or pulse laser illumination. And we can have the collective regime. So here it's more like a barbecue. You have many nanoparticles that are heating at the same time. Each nanoparticle can see the heat of each others. We have that many, many, many times because you have like more than thousands nanoparticles illuminated at the same time. So it's a temperature delocalization regime, and the temperature is uniform through the whole nanoparticles assembly, despite the nanosource, the heat source nanometric size. And it happens on substrate with high density of nanoparticles when we shine a light on a large area. And it happens with continuous wave laser illumination. So clearly, I got like micrometric polymer dots. My sample is looking like this. So clearly, I can do the estimation of the relative contribution of temperature generation, so between the confinement regime and the delocalization regime. And here I am in the regime of the barbecue. So the heat is generated through collective effect of nanoparticles. So that's why I have big dots of polymers. Knowing this, it's possible to perform numerical calculation to know the temperature that is reached by the samples. For that, you need to do some calculation, so to know the field, to know the absorption cross-section of the nanoparticles of the samples. And then you can plot heat here. So it's the blue curve here. So it's a theoretical curve. And on this figure, I added the first observation of polymer. So here it's the first power that I use for which I observe polymer. And here there is no polymerization. So the threshold on my sample is here. So it's at around 100 degrees, which is close to the experimental threshold of my formulation. So clearly here I graphed polymers through thermoplasmonics, but in a micrometric size. And what I wanted to do is to graft polymers, but locally at the nanometric size. And to do that, I need to control heat generation on the sample, because I don't want to have this regime anymore. I want to be in the confinement regime. And if I want to keep my laser, so continuous wave illumination, I will need to focus the laser to 20 nanometers, which is not possible. And another way to do that is to use the femtosecond pulse laser in order to have confinement regime inside my samples. So I access a femtosecond pulse laser thanks to the PEPR Luma and the platform ultrafast. So I did this experiment in Strasbourg at IPCMS. So here on the first line, I remind you what I observed with a continuous wave laser. And here is the behavior with femtosecond pulse laser with the same wavelength. So it's 532 nanometers, and it's like the same samples. And here we can see that the behavior is way different. We observed dark spots, which is not really high, which is not really big. If we observe the extinction of the particles, we observed a slight redshift of the plasmonic resonance, meaning that the refractive index was locally higher than here. So clearly here, we have polymer. But we don't know here how is the dispersion of polymer. Is it like polymer shell? Is it a polymer layer on the top of the particles? We don't know. So I did a simulation, because of course I tried to image these samples with atomic force microscopy, with profilometry, with scanning electron microscope, but each time I did not see anything. But it's due to the fact that 9 nanometer shift in the resonance wavelength correspond to only a 2 nanometer layer of polymer around my nanoparticles. So that's why it's difficult to image. So I performed the same experiment, but on 10 grids. So with 10 grids, I can access the nanometric resolution. And here I observe the 2 nanometer layer around the nanoparticles. So clearly, experimentally, I achieved to confine the heat. We can perform calculation to check that we are really in the confinement regime. The first one, which is really important here, is to check that the nanoparticles had the time to cool between 2 pulses, because it's a pulse illumination laser. So you have a pulse. The temperature of the nanoparticle will first increase, and then between the 2 pulses, it's important that the nanoparticle has time to cool fully between 2 pulses in order to avoid any accumulation of temperature. So clearly, here we are in the confinement regime. We can calculate the temperature, which is actually reached theoretically by the sample. And here again, the threshold is here. So here it's the power, the first power that is used to observe polymer on the sample. Here I observe no polymer, here I observe polymer. So the threshold is in between. So it corresponds to the experimental value here of 130 degrees. So here we have a strong correlation between theory and experiments. So here, with the same thermopolymer, we achieve to control the heating of the sample with the illumination. So for continuous wave laser illumination, we generate a collective heating, which is the barbecue. For femtosecond pulse laser illumination, we confine the heat inside the nanoparticles. So we are in the Shah-Wizard regime, also localized heating. And using the same temperature probe, we effectively observed this. So here we observed micrometric polymer dots, and here we observed nanometric polymer shell. So here I clearly designed a temperature probe at 130 degrees that can be used in thermoplasmonics at the macro scale and at the nanoscale. So why is it so interesting? So if we look back to the application of plasmonics, so we have many applications of plasmonics. And as soon as we have the plasmon, we have the free effects. Even if you only want to have like electron-hole pair, the plasmonic nanoparticle will generate heat. And if we take this example of photocatalysis, you want to have the chemical reactions that take place efficiently. You don't want to destroy heat by heating. So it's important to know at the nanoscale where the nanoparticles will heat the most in order to prevent any degradation of any chemical species that you don't want. So I did the same experiments that I showed you for photopolymerization, but with my thermopolymer. So I took the nanotriangles here on the time grids, which are colloidal. I put a droplet of my formulation and then illuminated it to see with a nanometric resolution where the heat is generated and and to quantify the temperature that is reached by the nanotriangle. And I observed this polymer distribution. So I observed polymer here at the tip of the triangle, which is corresponding to the polarization of the incident field. So here the polarization is along these two, and I observe polymer here and here. When the polarization is along only this tip, I observe only polymer here. And this was clearly not expected. The physician and the physicist and let's say the theoretical physicists, they expected to have an homogeneous polymer layer around the nanotriangles due to the fact that the thermal conductivity of gold and the thermal diffusivity of gold are both way higher than the one of the polymer. So the temperature should be homogeneous inside the nanoparticle before diffusing in the surrounding. So all of this community was expecting an homogeneous layer around the nanotriangle. In the second part, oops, sorry. So here, it's the result that I obtained with my thermal initiator. So here it's thermopolymerization. If I put back the results that I obtained in photopolymerization that I presented in the second part of my talk, it's like the same behavior. So we have polymers here where the field is the highest, corresponding to the thermopolymerization as well. But it's not the same formulation. This formulation cannot photopolymerize. Here it's thermic reactions that are driven by temperature. Here it's a reaction that is driven by light. So why do we observe that? So we try to do the same experiment, but with removing the thermal initiator. So this formulation is only made of monomer, but the monomer will at a certain temperature. So here it's highest temperature at 220 degrees. At some point, it will start to polymerize by itself. And we observed that the incident power needed to observe that is way higher than this one. So clearly here, it validates the role of the thermal initiator in the polymerization process. And in a way, it removes totally the photopolymerization pathway here. Moreover, if we send a power which is way higher, we observed the triangle which are melting, but they are not melting in an homogeneous way. They are melting from the tips that are excited here. And what we observed is that we observe also that the polymer around the nanoparticles here was present before the nanoparticle reconfiguration. So the polymerization was faster in terms of time scales than the reconfiguration of the nanoparticle, which was not expected at all as well. So in order to explain this experimental results, which is highly reproducible, I'm starting a collaboration with Jean-François Brich at LN2, who is able to perform electronic temperature map of the nanoparticles. So here it's not, let's say, real temperature, but it's electronic temperature. And we can observe that the electronic temperature can explain the polymer presence here at the tip of the particles. But it will, if the electronic temperature is at the source of the polymerization, it means that the reaction of polymerization is really fast, that it took place in less than 30 femtoseconds, which is like really, really small. And we cannot know here from this if the polymerization observed here is coming from only a thermal process or an electronic process as soon as it's electronic temperature. So as a conclusion here for plasmonic and chemistry, so we can use the field enhancement to photopolymerize and to put molecules where we want, where the field enhancement is the highest for sensing. We can use it to with thermopolymerization to know the temperature that is reached by the particles at the microscale and at the nanoscale, and we can use this as a tool for temperature mapping for single nanoparticles. So here, clearly, with these two chemistry, we can address selectively one of the other mechanisms. So here we want to map the field enhancement, and here we want to map the temperature. So it will lead to new fabrication process with thermal or photoresponsive materials that can be grafted at the nanoscale. So as a general conclusion, we have, let's say, a single plasmonic effect, but divided into field enhancement with, So with a single plasmonic effect, but we can observe, it can generate light and heat, and it can be used in chemistry and biology. So for the field enhancement, as I said before, we can map the field enhancements. With the heat, with biology, we were able to observe the growth of thermophiles. And with chemistry, we are now able to map the temperature at the nanoscale. So I want to thank the organizer for inviting me here. I want to thank all the partners with which I'm working on and which were funding this research. So Université de Haute-Alsace, CNRS, the ANR Popcorn, the PEP Luma and the Sunrise Project, and the Fondation de la Maison de la Chimie, and you for your attention. Thank you.
Thank you, Céline, for this bright and insightful presentation. It's now time for questions. Who wants to start by asking a question to Céline, please? I was quite sure.
Yeah, actually, thank you for your very interesting presentation. Okay, yeah, it's better now. So thank you for your very interesting presentation. I would like to come back on the last result that you showed about the electronic temperature. Can you explain what could be the mechanism in that case? So no heating, but just high electronic temperature. So what could happen?
It's like the molecules which are close to the surface will start to will see the vibration of electrons, and there will be a transfer of energy, which cannot be called real temperature, but it's look like this. It's like interaction between them, and then we will start the polymerization. But it's like an hypothesis now. But for a chemist, it's temperature, but not for physicists.
Just another question about the 200 degrees that you showed for so the maximum temperature of water under plasmonic excitation. Is 200 degrees theoretical maximum, or because you mentioned that you can reach a higher temperature than 100 degrees because of the nucleation site that we're missing, but is there any maximum theoretical value?
If you try to put the laser power more, we cannot observe any. If the temperature is higher than 200 degrees, it will start to form bubbles. So you cannot perform any biological experiments with bubbles in your media. So that's why it's an experimental limits.
Thank you for your nice presentation. I have a question, two questions on the thermal polymerization. So as you said, you are at maximum of 200 degrees. And with such small nanoparticles, are you sure yet that you don't melt the gold nanoparticle as the melting temperature of nanoparticle is way below the melting temperature of bulk material?
So the limitation of 200 degrees, it's only in water. If you put polymers on top, you can reach the temperature of the polymer. So I don't know if it answers all the questions. But...
And on this result also, you said, if I remember well, 2 nanometers of polymers.
Yeah.
Is there at what it just capping on the nanoparticle, or how many layers of monomer do you think you have?
I think the layer it's radicals polymerization, so we cannot control this, but we have many layers of monomers. But as soon as you have one thermal amorcer, initiator which will break, you will start to form many polymers. So you cannot have like 1 nanometer or less of polymers. It's difficult due to the free radical polymerization system. Okay. It's really hard to constrain less than 1 nanometer.
Okay, thank you.
Other question?
So I would like to come back on the thermophile bacteria that you showed and the swims. Are they sensitive just to a specific temperature, or do they follow some temperature gradients, or so how does it work?
It was something that we wanted to investigate, but it was really hard due to the swimming behavior of the bacteria. But clearly it starts when the temperature is the one that they need, but it's hard to know where they go, because we have like a small field of view, so we cannot know, but sometimes they go outside of the field of view, which is like, which means that they go in cold environments by themselves. But biologists, and I think that there is a paper on that, which create gradients along gradients of temperature along capillaries, and they observe that the bacteria at some points they go where the temperature is the best for them. But we did not observe that in our setup.
Thank you.
I have a question concerning this slide as well. The extension we are observing on the image correspond to, in your opinion, electron-phonon coupling length or something more related to an electron mean-free path? I mean, is electrons leaving the metallic environment and going to the polymer? Or is it something which is really some phonon at this scale or something like that?
I think it's more phonons because the media is outside the nanoparticle. It's a dielectric media, so it's really. I think it's really hard to imagine to have an electron that is ejected from the particles and behave in the monomer and try to find the thermal initiator and break it. I don't think that occurs. So it's more that phono-phono interaction, which will make the molecule vibrate and somehow create heat.
And to define a temperature around an edge like this, what is the minimal size, physical limit to define something as a temperature?
I think theoretically they can do that, but with the polymer we can expect to have like nanometric.
Because the electron mean free path in gold is around 30 or 40 nanometers, so it's larger than the scale you're observing there. So just
but not outside of the particles.
Outside of the particle, I have no idea. Okay.
Because here the time scale is really, really short. I mean, the anisotropy that is represented here in electronic temperature, it's like less than 30 femtoseconds. So it's really short, and really short to induce a reaction in the polymer as well.
Thank you. Other questions? So it's time to conclude this session. So let's thank again CELINE for this presentation.